Hi everyone, I hope you're enjoying the conference so far. My name is Lauren McKinnon and I'm a researcher at Brigham Young University, and I'd like to present to you a comprehensive analysis of the phylogenetic signal in ramp sequences in 211 vertebrates. And the big idea in this project was to see what ramp sequences can tell us about phylogeny. So first off, what is a ramp sequence? Well, a few years ago, researchers discovered that many times at the beginning of genes, rare codons that aren't used very often will cluster here at the beginning. And this is important because as a ribosome is translating the ramp sequence, when it gets to these rare codons, it will take longer to translate it because it has to wait longer for the tRNA molecule to associate with it because there are less of them because the codon is rare. And so in genes that are highly expressed, the ribosome will move slowly in this beginning region and then it will speed up after the ramp, which means that when many ribosomes are attaching to the same gene, it will prevent any ribosomal collisions and it will decrease translational errors. And it's estimated that ramp sequences occur in approximately 15% of genes, which is quite high. And a couple years ago, researchers in my lab developed an algorithm called Xramp. And this algorithm is able to identify ramp sequences in a single gene. The way that it does this is it calculates the relative codon efficiencies using the codon frequencies. So a codon that is used frequently would be translated more quickly than a codon that is used rarely. And after it calculates the relative codon efficiency for each codon, it estimates the speed of the ribosome at each location in the gene based on a mean of 9 codons. And it uses a 9 codon mean because that's the estimate of the length of a ribosome. So after it does that, this results in an estimate of how fast translation occurs at each index, at each location in the gene. And then when there's outlier regions at the beginning of the gene, so when translation occurs significantly more slow at the beginning, then we identify that as a ramp sequence. In this particular project, we wanted to know if we could use just the presence or absence of ramp sequences in orthologs to recover phylogenies. So what we did was we first downloaded species FASTA files from the NCBI database, and looking at their genomes, we identified any ramp sequences that might exist in all of their genes. Then we looked at each orthologous gene across the vertebrates, and we determined if a ramp sequence was present or absent in each of those genes. And doing this, we used parsimony and maximum likelihood to recover phylogenies using just the presence and absence of ramps. Then, in order to determine if those phylogenies were congruent with what we currently understand phylogenies to be, we compared it with the Open Tree of Life taxonomy, and we quantified that using the branch percent identity. So you can see in this chart that using just ramp sequences, we were able to recover around 70% of relationships, of species relationships in the Open Tree of Life. And this is very comparable to other genomic attributes that I've listed here. For example, codon inversion, amino acid version, and several of these word-based methods all recovered a similar amount of species relationships as the ramp sequences. So this shows that using just the presence and absence of ramp sequences in orthologs, we're able to recover many of the species relationships that are seen in the Open Tree of Life. We also investigated ramp sequences and if they have a use as a novel phylogenetic character. And we went ahead and scored each ramp sequence in each ortholog to determine if it was a good character. So for each ramp sequence, we mapped it to the Open Tree of Life taxonomy, and we counted the number of origin and loss events for that particular ramp sequence. And using this, we calculated the retention index. And a retention index of 1 means that the ramp is completely homologous, whereas a retention index of 0 means that it has no phylogenetic information. This chart shows that many ramp sequences do have phylogenetic information, although some of them are homoplagious, as you can see by the zeros here. We wanted to know if that's a good result, so we did 1,000 random permutations. We shuffled up the open tree of life 1,000 times and repeated this process. And here in these graphs, you can see that the 1,000 random permutations resulted in much lower retention indexes as did our actual, which is shown way off here to the right by this dotted line. So this shows that there's an empirical p-value of 0, and it shows that ramp sequences do contain phylogenetic information. So overall in this study, we learned that the presence and absence of a ramp sequence in many orthologs is largely congruent with the open tree of life. And we also learned that although some ramp sequences may be homoplagious, if you look at the overall usage of ramp sequences, then they do show statistically significant levels of homology as compared to the open tree of life. And all of this indicates that ramp sequences may be used as a novel character in future phylogenetic studies. Thank you, and I'd be happy to answer any of your questions if you contact me. Bye.